BioSentinel, a little life in infinite space. Cool science, so what? The BioSentinel mission was selected as one of the secondary payloads and the only biological experiment to fly on the first launch of the Space Launch System rocket for the Artemis I mission. The primary objective is to develop a biosensor instrument to detect and measure the impact of space radiation on living organisms beyond Earth's magnetosphere, getting us one step closer to being a multiplanetary species. Some background. Astronauts live in a pretty extreme environment aboard the International Space Station. The Earth's magnetic field shields space station crew from much of the radiation that can damage the DNA in our cells and lead to serious health problems. When future astronauts set off on long journeys deeper into space, they will be venturing into more perilous radiation environments and will need substantial protection. BioSentinel will perform the first long-duration biology experiment in deep space without the protection of Earth's magnetosphere. The hypothesis: can a small CubeSat be designed to controllably expose living yeast cells to deep space radiation conditions in a measurable and informative way? The mission. The primary function of BioSentinel is to monitor the vital signs of yeast to observe how they perform when exposed to deep space radiation. Because yeast cells have similar biological mechanisms to human cells, such as DNA damage and repair, studying yeast in space will help us better understand the risks of space radiation to humans and other biological organisms, as well as aid in the planning of crewed exploration missions to the moon and beyond. BioSentinel will specifically investigate yeast cell growth and metabolic activities after exposure to a high radiation environment beyond low Earth orbit. For BioSentinel, 2 yeast strains were created. One, a wild-type strain used as a control for DNA damage and yeast health. And 2, a DNA repair-deficient strain sensitive to ionizing radiation. Inside fluidic cards, both strains are loaded and dehydrated. Each payload comprises 18 fluidic cards with 16 microfluidic wells, for a total of 288 wells. Card activation happens at various periods over the 6-month mission and in response to ground orders. 2 fluidic cards are loaded into growth media containing nutrients and a metabolic indicator dye at each time point. In addition to card filling, the temperature of the cards rise from 4 degrees Celsius, which is stasis mode, to 23 degrees Celsius, which is cell development mode. When metabolically active yeast cells are present, the metabolic dye changes color from blue to pink and then pink to clear. A 3 LED optical detection device is used to monitor colorimetric changes in cell proliferation. The optical data reveals how quickly the cells can recover from radiation damage over time. Some limitations. Of course, yeast cells are not human beings, let alone human cells, but they are widely accepted primary models for human health studies, and there is still a lot this experiment can teach us about the true danger of deep space travel.